Thanks, Dave, I think. It's a pleasure to be here. I'd like to thank all the organizers for inviting me to give a chance to share some of the things my students and I have worked on over the past many years. Since we've got an hour, I actually changed the title to more broadly to non-invasive diagnostics, prenatal and otherwise, and we'll be talking about some things that happen after birth and are used to diagnose children for various maladies. And the theme that underlies it all will be, as David mentioned, this idea of trying to replace invasive biopsies with simple blood tests. And it's all oriented around the same sort of approach to measurement and the same sort of physiological principle, this notion of looking at DNA and RNA that circulates in the blood that's not part of any cell anymore, and marrying that phenomena with sort of the power of using next generation sequencing to analyze it. So quick slide on conflicts of interest. I've had the pleasure of seeing a lot of our work get translated into the clinic and have participated along the way. And so these are some of the companies that I have been and in some cases still am involved with. If I mention a company name in the course of the talk, chances are it's on this slide. The story starts when I got interested in prenatal diagnosis, and when I first became a parent, my wife and unborn daughter underwent amniocentesis, then our next child, CVS, and I realized this is kind of a crazy situation where the doctor is suggesting we get a diagnostic test, but there's risk of losing both the baby and potentially the mom simply as a result of asking a diagnostic question. And that just did not seem right. And so it sort of stuck in my head, is there some way to ask genetic questions of a baby before birth without having to do it invasively? And the challenge here, of course, is that this invasive sampling creates risk of miscarriage. The sort of most common disorder, a genetic disorder at live birth, is an aneuploidy, extra copy of a chromosome, and the risk of losing the baby is roughly comparable to the incidence here. So about 0.5% to 1% of the time, something really bad happens, which just didn't seem acceptable. And sort of the solution to this was actually discovered in the late 1940s, and it's this thing I mentioned, this phenomenon of cell-free DNA. And so these two fellows working in Strasbourg realized that there's DNA that circulates in the blood that's not part of the nucleus anymore. It's the detritus of dead cells. When a cell dies, it dumps its nucleus and its genome into the blood. The genome gets chewed up in little pieces, and that DNA kind of circulates there. This is the data from their paper, and they looked at people who were healthy and had various afflictions, one of which, if you look at the very last one, was pregnancy. The text underneath is what I clipped from the paper, and they commented, in the case of pregnancy, we see higher values, so something seemed to be going on. So even back then they noticed, and this was completely forgotten for decades. So this paper was lost in the literature, never cited. People sort of remembered that this phenomenon existed. There was a funny little corner of the oncology world that was trying to use it as a diagnostic, but it was kind of lost. And then kind of revisited again in the late 90s at Oxford, where Jim Wainscoat's group decided to look again at pregnancy. They had been working in oncology, and using molecular biology, which had been invented in the interim, they used PCR to show that some of the DNA in the blood of a pregnant woman comes from the fetus. And they did that by looking at women who were pregnant with male fetuses. They looked for fragments of the male sex chromosome, which should not ever be present in a woman's blood, and it turns out it's there only when they're pregnant with a male baby. So that set off a huge race to try to figure out how to use this as a way to ask questions about the genetics of the baby via a simple blood test. And it turns out these aneuploidies are really tough to do. It's not that there's some mutation that's different, but the genetics are normal. You just have an extra copy of one chromosome. So many of the sort of clever PCR tricks that people developed aren't applicable here. And there were a lot of false directions, and people went down wrong paths. And we were fortunate enough to work out the answer here at Stanford, which has now become the dominant way of approaching these sorts of questions. This is Christina Fan, who was my graduate student at the time. I did the work in my lab. And we worked out a way of sort of, in principle, using counting to solve this problem. And so much of the effort in the field had been oriented around trying to purify the fetal DNA and separate it from the mother's DNA. Because most of what's there is the mom's DNA. The fetal part is a small fraction, just a couple of percent. And so there's this huge background of normal DNA, and people thought, oh, if I could purify the fetal part, we could make a measurement. That proved to be impossible, and our method doesn't require you to distinguish maternal from fetal. The idea is simply to look for overrepresentation of one chromosome with respect to another. And so if you have some way of counting molecules and assigning which chromosome they come from, the sort of simple laws of statistics say that you should be able to do that for arbitrarily small differences simply by counting high enough. And by the basic laws of statistics, your error goes like 1 over square root of n. So the higher you count, the more precise your measurement. The challenge here is how would you count molecules? And that's where the other sort of ingredient comes in, is these new generation of DNA sequencers called next generation sequencing. Part of my research was trying to develop those, and we did it by analyzing single molecules. And the story sort of, for me, started in 2003, where we kind of first proved it worked on a very small number of molecules and a ridiculously small read length. Many arguments with the referees about whether it could be generalized. Turned out it could. Within a few years, the company that was developing the technology had brought the read length up to something much more interesting, a couple of dozen base pairs. They were doing millions of molecules at a time. And then they built an instrument that allowed you to do 1 billion molecules at a time. And this was the first sequencer that allowed you to sequence the human genome on a single instrument. And we did that, and we explored it as something sort of interesting for human health. But for the purpose of this talk, I don't want you to think about these machines as genome sequencers anymore. I want you to think about them as molecular counters, because that's how we're going to use them. And the point is that the way they work is taking lots of little fragments of DNA, spreading them over a surface, and sequencing them. All sequencers essentially work that way now. And the parallelism is enormous. So, we did 1 billion in 2009, as many billions now. And so it gives you the ability to really count lots of molecules and ask questions about their statistics and their relationships. And in particular, we realized this was a great way to solve that problem of measuring overrepresentation in fetal aneuploidies. So we teamed up with Yair Blumenfeld here in the EMFM group. He and his colleagues recruited a bunch of women to a study. They drew blood. They did independent invasive karyotyping. And then Christina purified the cell-free DNA from the samples, sequenced them on a sequencer, counting up to about 5 million at a time. Every single sequence read was mapped to the average human genome, the reference human genome, and allowed to vote for a chromosome. And what we looked for was overrepresentation. And what you see here in the figure is the overrepresentation of chromosome 21 relative to the other chromosomes. That's the one for Down syndrome. And the red data points are all represent samples drawn from women who were carrying babies with Down syndrome. The blue from normal fetuses. And you can see very clearly the separation. It really works. There's nothing special about chromosome 21 in the measurement here. You can ask the same question of all the chromosomes, and it turns out that the other two most common trisomies are 18 and 13, and we were able to observe them in our data set as well here. So this is all the chromosomes on the x-axis, overrepresentation on the y-axis. Right in the middle is chromosome 21. And as you look to the left, you'll see 18 and 13, and the outliers there are the samples of the women whose fetuses had those trisomies. So we published this in 2008. It's quite a while ago. So we've seen what's happened in the interim. And immediately after we published it, it created an enormous amount of excitement. It was replicated very quickly within a couple of months. And several clinical trials were launched, both in academia and in industry. They read out about 3 years later with cumulative results on thousands of women. And it all sort of worked the way we said it would, because square root of n is a pretty fundamental thing. And in around 2012, the commercial tests were launched. There's now more than half a dozen companies offering versions of the test, and the numbers grew enormously. In the first year, it was 500,000, then doubled to 1 million. In recent years, it's somewhere around 3 million women a year. And in places where people have the choice, there's been measurements and academic studies done showing that the rates of use of amniocentesis and other invasive measurements are declining precipitously, as one would expect. It's really kind of a no-brainer. So that's been amazing to see. And if you imagine even a fraction of these women are avoiding invasive tests, that amounts to many thousands of lives being saved every year simply by making a better diagnostic. So while I wasn't involved in the clinical trials or all that, and so in my lab we were thinking about other ways to use this counting principle as that was going on. And we began to wonder if we could ask other questions about the fetal genome. For example, could we detect structural variations like deletions in the genome? And those cause a certain amount of genetic disease as well. A very famous one is DiGeorge syndrome, which is a 3 megabase pair deletion, the vast majority of which are de novo deletions. So it's not like the parents there's an inheritance problem with the parents, and you can't carry your screen for it. So we ended up having a case come through the clinic here at Stanford, and they were recruited to the study. And so we were able to look to see if we could detect not a whole chromosome changing, but a small fraction of a chromosome. And so if you think about this counting principle, remember I said you're mapping to the genome, you're voting for a chromosome, but you could change the voting rules. You could say, we're going to divide each chromosome up into bins, and you can vote for not just the chromosome, but a particular bin in the chromosome. We can change the size of the bins. And so that's what we did here to look for structural variation. So now we're looking across the whole genome here, and you can see the relative representation from this sample. And if you look down at chromosome 22, you'll see a dip there. If we zoom in on that, you can see right in the region where the DiGeorge deletion happens. In this case, there is a really substantial dip in the relative representation of bins, and the statistical significance for that is really quite substantial, and it's right exactly where it's supposed to be. And so this shows how you can use this sort of counting approach to move beyond aneuploidies to look at structural variations in the genome. But why stop there? I mean, what really limits the size of the bin you can use? There's nothing fundamental that limits it. You can imagine making the bin smaller and smaller and smaller until it's just one base pair wide. And at that point, you're able to read out any given point mutation in the fetal genome. And so this counting principle should apply at the level of individual single nucleotide polymorphisms and mutations. And so we decided to see if that was true as well. And initially, we did this a couple of different ways. Just for simplicity, I'll talk about the exome, which is the coding part of the genome, so the part that codes for protein. It represents about 1% of the genome and is usually the most informative as far as these things go. So we did a pull down of the exome, purified that part of the genome, sequenced it at high depth, and Wei Gu, who's a PhD MD student at the time, is the one who did that work. And he was able to show that, in fact, you can read out any coding mutation you wanted to in the fetal genome. And as we showed with the aneuploidies, the higher you counted, the deeper you sequenced, the better the measurement got. And this kind of completes the story. And you could ask essentially any question you want about the genetics of the fetus from a simple blood draw from the mom. And this has obvious applications to some of the things you heard earlier this morning, in particular the SMA story. The other thing we were working on in the lab was other ways to do biopsy replacement. And when we published the first paper, word got around Stanford that this was something I was interested in. I got a call one day from Hannah Valentine, who was in the cardiology department, and she said, when we do heart transplants, we have to go biopsy the patients serially for quite a bit after the operation to make sure they're not rejecting the heart. And it would be nice to be able to do that with a blood test rather than the biopsy. And so I began to think about whether we could use these ideas in that context as well. And it turns out there's a very simple way to do that, and it's to think about the heart transplant as a genome transplant. And this is what I mean by that. So imagine before you get someone else's heart, you have all your organs that you were born with, and they all have the same genome. Every cell and every tissue and organ has the same genome. It's your genome. We'll call that a red genome. So all the cell-free DNA in your blood is red. Now imagine you get a heart transplant. Someone else's heart is put in your body. Every cell of that heart has a different genome. It's got the donor's genome. So now, whenever a cell from that heart dies, you get a little bit of green DNA in your blood. And now you have a mixture of green and red DNA in your blood, and the green ones are the ones from the heart. And they're different. They have different polymorphisms. So imagine your immune system starts to attack the heart because you're rejecting the transplant. More cells will die, more green DNA will come in the blood, and the relative amounts will change. And so by measuring the proportion of green to red DNA, you can figure out whether this individual is having a rejection event or has successfully received the heart. So it's a pretty simple idea, and it's pretty straightforward to do using the methods I just described, except in this case, we're not counting against the reference genome. We're sequencing each little fragment and looking at the polymorphisms that are there and separating informatically separating the donor's polymorphisms from the recipient's. And so we look for things that are not part of the recipient genome, and we start counting those. Each molecule that has a polymorphism that's not part of the recipient's genome, we put a little click, and that's what's shown here in these graphs. That's Hannah Valentine, as well as my former postdoc, Tom Snyder, who did the work. So these are 5 different patients. The x-axis is months time, months post-transplant. The y-axis is that percentage of donor DNA. The blue curves are the one to look at. The green ones are an estimate of error. And you can see at every single time point here, these patients were biopsied. And some pathologists looked at the biopsy and said rejecting, not rejecting. The places where the pathologist said rejecting are shown with red arrows. And you can see they correspond very clearly with an increase in donor DNA. So it all sort of works the way you think it would. And in fact, it appears that this DNA-based measurement looks to be more sensitive because it's increasing before the pathologist starts to see anything. So that got us very excited. That was kind of a proof of principle. And we published that description on a relatively small number of samples. And I'll just point out that we use the word universal because there's nothing in what I've described about this that depends at all on the physiology of the heart as an organ, right? I mean, this should work for any transplanted organ. And so we ended up getting a large grant from the NIH to look at heart and lung transplant patients. And for 3 years, virtually every heart and lung transplant patient at Stanford was enrolled in our study. We followed them longitudinally. In the middle of it, Hannah left to take a leadership position at the NIH, and her colleague Kiran Kush took over. We were able to sort of validate this approach on a pretty large number of samples and individuals. This is more than 500 samples, and a third of the patients here were pediatric. Okay, and the age ranged from 17 down to 2 years old, and you can imagine it's a big deal for any kid not to have a biopsy. I mean, it's hard to draw blood, as we heard earlier, even harder to get a actual biopsy out. And so, but I had a chance to meet some of the kids and patients. They're all super grateful that there was a prospect of getting rid of these biopsies. And it worked really pretty well. So this receiver-operator curve shows the performance. There's a bunch of different comparisons being done there. If you look at the solid line, the darkest one, which is the overall comparison of the blood test to invasive biopsy, the area under the curve is about 0.8. So that's pretty good. And you would frankly have been astonished it was anything better than that. It would mean that there's something wrong, because the biopsy is, even though it's the gold standard, it's simply because there had been nothing else. It's actually not such a great measurement. And that's been known in the community. People have done studies when they have separate independent pathologists look at the same slide. The concordance rate is something like 75%, right? And so even if you had a perfect diagnostic, and you're trying to compare it to the biopsies, you'd never do better than this. And so we ended up, in the cases where we disagreed with biopsy, going back and looking for ways to referee that. And we had independent things to look at in the clinical record. We had the longitudinal information. And most of the time, the resolution went the way of the blood test. So we really do think it's superior to biopsy in its performance, because it's integrating over the whole organ. among other things. In the lung cohort, also with a number of children involved, it works equally well, both compared to biopsy and other physiological measures of lung dysfunction, which are shown here in a variety of different metrics. I won't dwell on all the details, but we worked on this in collaboration with Dave Weil, Dave Cornfield, and Mark Nichols. And this shows that it works in other tissues as one would have expected. So this is now in the clinic through a company called Childx, and they licensed the technology. They decided they wanted to first apply it in kidney and did a number of studies in kidney, and that's now out there both as a product and something getting reimbursed by Medicare. And they just last week launched it in heart in conjunction with their prior gene expression-based test. And so it's something that is affecting the lives of patients, hopefully in a positive way. When we were doing the large study here at Stanford, my postdoc, Ewan de Vlamink, came to me one day and said, you know, when I look at the data coming off the sequencer, about 99% of the reads are mapping to the human genome. So not everything was mapping to the human genome. And I said, well, that's great, 98%. That's pretty good. That means, you know, we must have very low levels of contamination and it's all kind of working. But he was sort of stuck on that and looked a little closer, and he realized that 1 or 2% of reads that weren't mapping to the human genome were real, and they were non-human, and they were a reflection of the microbiomes of the patients. So it's not just human DNA that's circulating in the blood, it's viral and bacterial and fungal and things like that. And he went and did a very sort of detailed analysis of that, and sort of broke down the whole representation that's shown here on this figure at various degrees of phylogenetic specificity. If you look at the center little pie chart, that's the coarsest. And on a kind of per genome basis, about 3/4 of what's there is viral, about 1/4 is bacterial, and a tiny little sliver is eukaryotic or fungal. And we realized that we had a really interesting opportunity based on a cohort we were studying who were transplant patients to ask what happens to your microbiome when your immune system is turned off. All these patients were getting drugs that were turning down their immune system, and the doctors would monitor that really closely. It's a crucial thing in the treatment to turn down the immune system enough that the body doesn't reject the new organ, but not so much that the patients are getting infectious disease all the time. So they watch that very carefully and collect good data on that. And so we decided to look at this cohort and look at in this case, their virome as a function of how much immunosuppression or other drugs they were receiving. And this slide gives you kind of a key for how to think about it going forward and what some of the key questions are. Herpes viruses, which are going to be the dark blue slice of the pie, are a very known important issue in transplantation because cytomegalovirus, CMV, which is part of that family, is known to cause rejection. So they watch that very, very carefully. If there's any whiff of CMV around, the patients are immediately put on antivirals prophylactically. Polyomavirus, which is the red slice, those cause cancer. You suppress the immune system, it's going to increase their activity, and sure enough, all these patients have increased rates of cancer in part due to that effect. Adenoviruses, which are green slice, they cause respiratory infection, and again, that's the flip side of too much immunosuppression. These things come in and make people sick. And the orange slice, which is probably the least familiar, are anelloviruses. These are a sort of mysterious group. They're ubiquitous among humans. They're not known to be a pathogen, but we all sort of have them. The tropism isn't really known, but they play an important part of the story going forward. And here's the punchline. This is Ewan, the postdoc I mentioned. So these pie charts represent the virome averaged over many patients at a given level of immunosuppression and antiviral drugs. So the upper left-hand corner is the least immunosuppressed and no immunosuppression, no antiviral. The bottom right is both of them all cranked up. And underneath Ewan there are healthy controls, one of which are pregnant women. And the other of which are the transplant patients immediately the day after transplantation and before any of the drugs have had a chance to kick in. And so what you see as you look at this is as you turn down the immune system, the yellow slice increases. Or as you knock out the herpes viruses, the yellow slice increases. So those, I'll remind you, are the viruses, the mysterious ones, and they seem to be in the most intimate dance with our immune system. So if you create an opening, either by turning off the immune system or by taking out a competitor virus, these are the ones that fill in. You can look at it in other ways. Here now is if you unfold the pie chart and look at it over time. So each slice here is a different pie chart, and this is time post-transplant. You can see that that as the immunosuppressants are kicking in, the viruses take over, and then as their drugs are tapered later on, 9 months after the surgery, they get rolled back again because the immune system is getting them under control. What about the bacteria? Well, those turn out to be pretty stable to the drugs. I mean, the bacteria are not being really controlled by the immune system the same way that the viruses are. Most of them are commensals, and they're sort of living in happy coexistence with us. And you can see that the phyla aren't changing as the course of drugs is administered and tapered. I've been showing you the relative units. There's absolute units as well. So here now, as the same data shown in absolute units and in genome equivalents. And you can see that it's not just the relative amount of virus that's changing, it's the absolute amount as well. Similarly, if you look at patients that have only immunosuppressants, the virus grow much faster than everything else. So that suggests, this is sort of interesting science, and but it also immediately suggests that we should be using this sort of phenomena to diagnose infectious disease, right? I mean, this could be a blood test that is hypothesis-free. It doesn't require you to order a particular growth condition or have a particular choice of primers to have a guess for what's causing an infection. You can, in principle, test for all infections at the same time. All DNA-based infections. It's not covering RNA viruses yet, but that'll come. So one of my postdocs, Mickey Cortez, decided to take that thought and run with it. He founded a company called Childx, which has created a test to test for infectious disease in a hypothesis-free manner. And you can see that it's a huge problem. Infectious disease, #2 cause of death worldwide. In the U.S., every year, there's more than 1 million patients who have an unresolved infectious disease where they can't diagnose it, right? And that's a little scary. And it sort of emphasizes the need for this. And again, this also connects with the theme of being a biopsy replacement. There's a certain number of infections that happen deep in the body, and if you want to sample it, you'd have to do a biopsy to get the material out. That's not always desirable or feasible. So Childx has built a test based on this. There are now, I mean, it's more than 1,000 different pathogens they'll detect for every single test, and they're able to turn it around in the course of a day. So it's being explored as a very powerful replacement for culture and other forms of molecular testing. Let me give you a few examples of some of the things they found. That have sort of flavor of the pediatric emphasis on it. So here was a 3-year-old girl who came in with pneumonia. She had been previously healthy, immunized, but had a number of problems: fever, cough, shortness of breath. The sort of PCR panel found her positive for viruses. Blood cultures were all negative. She was on a variety of antibiotics. They ran the test, came back as Streptococcus pneumoniae, a pretty common sort of pathogen, right? And it's kind of shocking that the conventional tests don't find it, but it's another case where there's a gold standard for testing, but it's not a perfect test by any means, and so there's plenty of room for improvement. So in this case, they were able to choose the appropriate antibiotic and narrow it down and help this young girl. Here's another one, 5-year-old girl with cancer. She had leukemia, and it's a cancer of the immune system, so she's immunosuppressed. And this is, again, a very common problem. I think we heard earlier talk about treating cancer with cellular therapy, that when the immune system has been attacked, you're more vulnerable to infectious disease, and it's a very common sort of thing the doctors are wrestling with. In this case, this girl came in with fever, neutropenia. Blood cultures were positive for Klebsiella. She was treated. Antibiotic, she improved, then the fevers came back. In this case, many things were tested for. Blood cultures for both bacteria, for fungi, PCR panels were done, all sorts of things were done. Everything was coming back negative. And so it was quite a mystery. The imaging looked terrible. There was something growing there in her lungs, and they ultimately did the Carious blood test, and it came back identifying the specific fungus that she had, and so she will be treated with an antifungal that helped for that. Here's the final one. This was a 9-year-old boy with endocarditis, and he had a history of heart disease, had a valve replacement. He came in with problems with fever and all sorts of other things that just really don't sound good. They started him on antibiotics. The blood cultures were negative, but they kept it going because, they were guessing that it was a bacterial infection affecting the heart. And, they found from imaging that the valve was getting destroyed. They switched around the drugs. Blood cultures were negative, and they switched the drugs again. Eventually, all the other tests they did, and this is sort of a common thing, you do one test after another, they're coming up negative. If you test for everything all at once, you can do one test. And what was found here was something called the HASEC group, which are bacteria known to be a cause of endocarditis, and it led to the choice of a very specific antibiotic to treat this particular bug. So, and actually, in this case, they were able to confirm that this was actually the correct diagnosis because they ended up having to take out the valve and were able to PCR directly from the tissue and show that it was the causative organism. So, as you sort of, that they've been doing this, these stories just sort of accumulate, and it's really quite amazing. And I can show you some of the data from about 500 different tests they've done, many of which were on pediatric cases, and about half of them were pediatric. And they covered a couple hundred different pathogens. The numbers are quite staggering, and the sort of top cases are people who are like the ones we saw: immunocompromised patients, people with fever, sepsis, things that are failing culture for one reason or another, or complicated, and all other diagnostics have not been informative. The most common things they're finding, the most common pathogens among those tests are shown here. Some maybe not surprising, some might surprise you. They also compiled a list of the ones they think are most interesting if you're if you're an infectious disease doc, things that you maybe wouldn't have thought to test for, things that aren't commonly tested for. When you do this hypothesis-free approach, you're really it's amazing what you find. So that story continues. I'd say their clinical evidence is accumulating that this is really going to be the future of infectious disease diagnostics. Carious is starting to publish their results. There's a number of registered trials ongoing, and it's all looks very promising to be a major transformation in how infectious disease is diagnosed. So in the last little part of the talk, I'd like to return back to that 1948 paper where I flashed up the data very quickly, where they discovered cell-free DNA. And if you had sharp eyes and were looking at the columns of the data, you'll see that they not only measured DNA in the blood, they also measured RNA in the blood. It turns out that there's also cell-free RNA that floats in the blood, and even though it's a delicate flower of a molecule, some bit of it survives, probably wrapped on particles, protected somehow, but is there. And that had kind of been ignored for a long time, and we decided some years ago to kind of take a full-on omic approach to it and do a transcriptomic analysis of what's floating around in the blood. And we did it returning to our favorite sort of model system of human development. And it turns out that we were able to see transcripts from the placenta and various fetal tissues floating in the mom's blood. And we wanted to try to use this as a window into human development and watch the whole gene expression program of human life unfold in front of us. And we did this as part of a phenomenal collaboration here at Stanford. Dave Stevenson and Gary Shaw, who've been put together the March of Dimes Prematurity Research Center, have organized a very large group of faculty, and we've been part of that. And it's been a really fun journey. We've also been collaborating with Mads Melby, who runs the Danish National Biobank, and has been interested in this sort of phenomena as well. And I'm going to describe for you some work that's been done in my lab by Tweenago and Mira Mufarej. And let's sort of start with this first question of trying to monitor human development. So Mads was able to do this amazing recruitment where he found a number of women who were very brave and were willing to come in every week during pregnancy and give blood. So very high density sampling throughout the course of pregnancy. And that's what's shown here on this. And we looked at about 30 of these women. And those are the folks in his lab who have been doing the recruitment and the collection. And we've been analyzing each of those samples by doing PCR on a large panel of genes. And it sort of gives us a transcriptomic clock of human development. So each panel here is a different gene, and they're color-coded. The orange are our placental genes, green are maternal immune genes, and purple are from various fetal tissues. And this represents an average over all the patients, and you can see over time that there's a variety of interesting trajectories. The placental genes are generally increasing with time. They go away after delivery. Some of them are going faster than others. Some plateau. The fetal genes tend to come up a little later. Some of them are plateauing earlier. The immune genes are also changing and have different baselines than the fetal and the placental. And you can see where this could be a very powerful tool to understand when things are potentially going wrong in development. There's a number of surgical techniques that are being developed now to correct in utero. There's going to be pharmacologic approaches too, and this is hopefully going to be the way such things are detected and measured in the future. We, as just sort of a simple application of this, showed that you could use it to demonstrate gestational age. And so by using machine learning on this, we were able to, from any given time point, predict reasonably well what the gestational age of the fetus was. And in fact, we did it. We showed that you don't need all the genes in the panel. 9 of them capture most of the information. Some of them relate to the biology in the way you'd expect. And it's roughly as good as ultrasound, which is kind of the gold standard here. Ultrasound works pretty well, but only in the first trimester. And so if you've missed that, this is a way to get the information of gestational age, or if you're in a resource-challenged area where it's just not feasible to have an ultrasound machine, which is a rather expensive device, you can, with a simple PCR test, get the same information. So we thought that was pretty cool. We also then began to think about, well, what the point of the center is, and what we've been kind of thinking about all along was, can you use this sort of approach not just to see development, but to predict when things are going to go wrong, like premature birth. And the reason why this center was formed is this is a really substantial problem. In the US, it's almost 400,000 babies each year. About 10% of pregnancies are affected, and it's the leading cause of neonatal death and complications later in life. And there's no way to predict it. I mean, it's really kind of an open greenfields area. So we, through the center and collaborations that Gary and David helped set up, were able to get access to some cohorts that had pretty high rates of preterm delivery. This one from University of Pennsylvania was collected by Mikhail Elovitz. And you can see here, it's not as densely sampled as our Danish one, but there's blood drawn somewhere around week 24 in postpartum, and the triangles show delivery. And you can see that this group were very, very early delivery. And that's sort of the most hazardous situation. So we use those samples to sequence the cell-free RNA and do gene discovery to figure out if there's anything that predicted preterm birth. And then we had a second cohort from the University of Alabama, collected by Joe Biggio, which we used as our validation cohort. So in the Penn cohort, we discovered about 38 genes that seem to do a pretty good job of separating preterm and full-term pregnancy. And you can see that when you sort of sort things based on those genes, it does a very good job of separating from the preterm and the full-term groups. We validated things with the PCR. That's probably not so interesting. And from that, we're able to show that not only when we do the PCR measurement of sort of 7 most interesting genes, it shows elevated levels in the affected cases of the Penn cohort, not in the unaffected cases. And in the Danish cases, all of whom are full term, it's sort of independently shows that they fall in the normal category. When we look at the independent cohort. Sorry, let me just skip ahead. It added in the Alabama cohort, which was not used for the discovery. They're now the ones marked with the Roman numeral 3. You can see that even on a single gene basis, there's an effect. And when you use them as a panel, it works reasonably well. Area under the curves of about 0.8 or so. I mean, it must be said this is a very small number of samples, so we're not claiming that it's ready to go as a diagnostic. But as a proof of principle, it's given us the confidence to set out to try to do a much larger prospective study. And we're very excited about where that's going to lead. And if it holds up, as from what we learned on this small data set, we should be able to predict spontaneous preterm birth about 2 months in advance of delivery. So I think I'll stop there and just sort of summarize. We have had quite an amazing journey over the past decade here, and it's been just a fantastic collaboration between my group, which primarily works on basic science and technology development, and a number of different clinicians here at the hospital. And none of this, would have happened had I not moved to Stanford 12 years ago. So I'm very grateful for that. And it was certainly a new direction for my research. So I've taken you through examples of non-invasive tests for prenatal genetic disorders, for non-genetic things, physiological disorders like preterm birth, showed how the same technology can be used in very different areas such as transplantation rejection and infectious disease. And it's just been absolutely sort of marvelous to see this sort of the breadth of application of this approach and how it's helping improve people's lives, children and adult. Thank you.
Well, thank you, Dr. Quake. That was a tremendous discussion, and I think that a good percentage of the audience is going to have some questions. But let me just ask the very first question while we get the assembly of electronic questions, and that is, you've shown us what's happened, and you've actually given us a nice eye into what might be the future. Could you kind of share what you think's next?
What's next? Well,
because this is all old stuff now.
Yeah,
we're
it's all very old.
Yeah,
exactly. I didn't talk about oncology. That's another area where this has been continuously investigated for decades now, and the folks in that field have adopted the approach we developed by marrying the Cellfree DNA to sequencing, and there's a number of tests already on the market, others coming out, and so you're going to see it play an important role in management of cancer. That's, the clinical studies to validate it are still happening, but it would be shocking to me if it doesn't prove useful in that context. What else is coming down the road? I think these tests, as they are more broadly adopted, are going to be an important driver of equity in healthcare, actually, because the biopsies require you to have tertiary care, a Stanford-type hospital with very skilled physicians. And if you don't happen to live within an hour's drive of Stanford, you're not, or UCSF or wherever, you're not having access to that kind of care. And whereas you can draw blood anywhere and mail it in. And so I think as these tests are more broadly used, it's going to really level the playing field of sort of healthcare across the country.
Great. So in some ways, you're almost arguing for a more egalitarian approach, almost like how we see the smartphone has created more access for all sorts of technology across all swaths of the socioeconomic ladder. So
that's right.
That's a very interesting approach. One question that just came in is, when do you anticipate widespread practice change to using blood samples instead of biopsies relative to transplant patients?
Oh, for transplantation, yeah. So, it's just happening in real time. I kind of showed the dates quickly. But, the kidney one was just launched last year, the heart last week. And so it's now a question of how quickly it's going to be taken up. It's there for people to use if they choose.
Dr. Quake, what are the cost-benefit analyses on the KRAS test versus the cultures and PCR tests that are done currently?
Yeah, that's an interesting one. Any given sort of culture or PCR test is not that expensive, but when you're doing 10, 15, 20, you saw them stack up, it adds up to quite a bit. And so what Mickey had worked out was, a single test actually does have a substantial cost-benefit advantage against many inexpensive tests that are done serially. And that's just kind of the first level of analysis. I mean, the other level is the sooner you catch, is the delay that it takes to get to the diagnosis. The sooner you get it, the sooner you can treat the patient with the appropriate drug, less time in the hospital and things like that. So there's all sorts of costs there that are incurred by late diagnosis that are hopefully going to be ameliorated by a hypothesis-free test. And that's, will be hopefully shown in the course of clinical studies over time.
Steve, one question that I have for you. You came here from Caltech largely because you wanted to have the benefit of collaborating with clinicians and doing translational science. And it's been obviously hugely successful. What do you think are sort of the ideal, what's part of the broth here? What's part of the DNA of the institution, if you will, that's allowed for that? And what part of that DNA do you think needs to be edited, so to speak, to increase this collaborative culture so that we can move forward still faster and further?
Yeah, well, my lab is in the Clark Center, the BioX building, and the essential part of the DNA seems to be the Pete's coffee there, because my lab is right next to it, and all the docs come there to get their coffee, and so there's a lot of serendipitous interactions just there in the coffee shop, and that's been really great. But sort of more generally, the fact that the medical school and the hospital is right here on campus, integrated with basic science and engineering, that's actually a pretty unusual thing. Not many universities have that. And they're often on different campuses, and that creates barriers. The fact that we're all here together, both professionally and personally, as you described in the introduction, I've done a fair amount of sort of scientific discussion, dropping my kids off at school in the morning over the years, and it's those contacts are really something special at Stanford.
Great. One other question that just rolled in is: can omics used to better understand the risk for spontaneous abortion in women and to better assess the risk associated with medicines or biologics?
Yeah, that's a really interesting question. So, people have done studies of spontaneous abortion, and it turns out, I think something like half of them can be explained by aneuploidy. So the vast majority of aneuploidies aren't survivable. 13, 18, 21 are, but you get one of the other ones, spontaneous abortion. And so we can, in principle, detect that early on with a blood test and give people a choice between surgical or pharmacological treatment. And that's never been demonstrated, that's been the thought, but in principle, that could happen.
And what about viral diagnostics in the non-immune compromised hosts? Is that possible?
Absolutely. Yeah. No, there's nothing about the immune suppression that was needed there. And Carius is diagnosing various viral infections. We've seen it in some of our pregnant women as well, a parvovirus at one point.
So one question relates to chronic fatigue syndrome that someone just asked, but a more broad question. Any questions that you have that you think if you had clinical substrate for that you could answer straight away?
Yeah, I mean, chronic fatigue is a tough one, right? And we did spend some time looking at that, fruitlessly, unfortunately. We're open to suggestions. Let me put it that way.
So that just brings up a comment that I'd like to make. And so there are a ton of outstanding basic scientists around who are so interested in clinical interactions. And I would encourage any clinicians here that have a hankering to get engaged with some of the basic scientists to really do so, because people like Dr. Quake are dying for a clinical substrate, so to speak. So I think that would be something that you could really take advantage of, such as we've seen in the course of this talk. What do you think? Can you give us amniocentesis, expensive, CVS, risky? Can you give us a cost comparison of the two modalities versus cell-free DNA? Yeah, that's the other cool thing about these tests is there's a very strong health economic argument because biopsies are expensive, broadly speaking. It's $3,000 to $5,000 for any kind of biopsy, whether it's for transplantation, for amniocentesis, whatever. And so that's kind of the baseline.
And the prenatal tests were initially priced to be about the same as the invasive tests, and then there was so much competition in the field that the price started dropping, and so now some of them are a tenth as much as the amniocentesis. So it's a very virtuous cycle.
The stakes are very high, especially relative to prenatal diagnosis. False positive rate, false negative rate, ascertainment bias.
Yeah. So it again gets back to how good is the gold standard, okay? And amniocentesis, while very good, is not perfect. It's got a false positive and false negative rate, and those are somewhere between a tenth and 1%, something like that each. It also has a fatality rate, as I mentioned, of about close to 1%. And so, if you want a single metric or statistic, I would call it the probability of undesired outcome. The false positives plus the false negatives plus the fatalities. And if you compare amniocentesis to the cell-free DNA-based test, cell-free DNA is winning by quite a bit by now, actually, the best ones. They're not all the same. That's an important point. So there's been this, the different companies have taken different tactics. Some have said, we want it to be as good as possible. We want it to be diagnostic quality. It's going to replace amniocentesis. And they've built products that are that good. And they tend to be a little more expensive. Others have said, we want to offer the cheapest product. We want to use it like a screen and use it to kind of send people in for amnio or not. And we'll accept worse performance because we're going to compete on cost. And so you got to bear that in mind. They're not all the same test, and based on what you want, you need to choose the one that's most interesting to you. But at the very high end, the best ones are better than amnio right now. And there's been tens of thousands of women's studies now that validate that.
So I'm going to get a little bit personal with you, but it's really not that personal because it's been, I think it was published in Nature, your personal genome.
Yep.
And Steve, I'll let him comment to that. But I'd like you to comment on how that happened, and also I'd like you to comment on your decision not to use a statin in that context.
I'm revisiting that one, actually. As I sort of mentioned earlier in the talk, we had helped build a sequencer, and we thought it would be a very powerful thing to use for human genome sequencing. That was just emerging. The costs were millions of dollars per genome, and the company was called Helicos that built the instrument I invented. There was a lot of uncertainty, controversy. Was the sequence quality good enough to do a whole human genome, and the read length's long enough? And so we decided we'd just do it. And so we did my genome, and it was one of the first handful of human genomes ever sequenced, and we published that in 2008, 2009. And then, what to do with it? How do you interpret it? The software was very rudimentary, and I was kind of tinkering around with trying to understand what it would mean for my personal health. And one day, you and Ashley came into my office to discuss something, and he said, "What's that on your screen?" I said, "That's my genome." And he was like, "What?" And I said, "Yeah, what is it? What is it? How is it going to help you treat me?" And so that marked the beginning of a very fun collaboration where a bunch of sort of put upon colleagues here in the medical school had to answer the question of how do you treat a patient who walks into their office with a genome in hand. And
very Silicon Valley.
Exactly. And being very opportunistic academics, they turned it into a lovely paper in the Lancet, which it answered that question. It was the first clinical annotation of a human genome, so I was the patient for that. But also a co-author. It drove the genetic counselors nuts, because that's not really the way it's supposed to work. But I was in the unique position of having the best understanding of how good the quality of the genome was, and where were sequencing errors, and what were things likely to be wrong versus correct, and so forth.
And you probably really needed the publication too.
And that was a fun collaboration, and it I think provided the sort of the nucleus or the genesis of what is now a sort of very thriving program here to use the genome in clinical medicine that a number of faculty are involved with in the hospital.
And the statin?
Yeah, so I'm big on the NNT, and number needed to treat, and for statins, no other background, it's like 100 or something, right? And so why would I start the statins now, even though my cholesterol was a little bit high? Now my cholesterol is a lot high, so they're really angry with me, and I'm starting to think I'm going to have to relent on that. No matter how much I exercise, it just keeps going up.
I understand that I think wine drinking can be
Must be great for it. Yeah,
can be helpful.
Right, exactly. I'm working on that.
So you've changed a lot of different paradigms, and have you experienced a lot of resistance along the way in any of these spheres? What frustrations have you had? What barriers have you had to overcome in getting some of these things adopted?
Yeah, I mean, it's mostly been the opposite, actually. I mean, we published the first paper on the fetal aneuploidy detection. You know, it got some press, and the reporters asked me, when do you think this will be in the clinic? I said, I don't know, 10 years? I mean, this is a tough thing to do. You got to do all these big studies, and it happened in 3 years. I mean, it was really accelerated. And, you know, the only people who were happy with the existing state of affairs were the pathologists who read out the karyotypes, right? They were seeing a bit of loss of business, and so they resisted a little bit, but they were quickly swept aside by the patients, the doctors. I mean, it just, everyone was looking for something better. And you know, I have to say the companies who were developing it were quite responsible in that, you know, they engaged with the scientific literature, they published their results in peer-reviewed journals, data was available, and that helped the medical professional societies kind of get on board that it really was working as claimed.
Wonderful. So one other question, which I think probably merits some comment, is so what do you think is the role of the university relative to the role of a company? In the abstract, Jim Woody talked about this a little bit. You obviously have some expertise as well, and it'd be great to hear your thoughts.
Yeah. So you know, they're both valuable, and they work together. In my experience, if you do it right, and you know, the things that you know, are best done in the university context are kind of the early discovery, the proof of principle, when you know you need the patience, the inspiration, the spark. I mean, and sort of the first demonstration that it's going to work, the proof of principle. And when it's time to, you know, make it bigger by either, you know, becoming an instrument that anybody can use, or clinical trial that requires, you know, large group of patients, big interdisciplinary teams, that sort of thing. Those things I find work better in the company context. You know, it's actually paradoxically, I think, easier to do a large multidisciplinary project in a company because nobody's worried about who's going to be the last author on the paper, right? They're all very aligned in trying to make a useful product that'll help human health, and so some of that stuff goes away in that context, and it also allows for a very singular focus, and that's important in getting something big across the finish line.
Great. And the final question I have, what book are you reading currently that you think people should give some thought to?
What book am I reading? You know, I just finished reading James Gleick's book on the information. That was fun. And after that, what did I read? Someone gave me Marcus Aurelius Meditations. I read that last week on my kids' spring break, and that was fun. Yeah, I'd recommend it.
Well, thank you, Professor Quake. That was a tremendous tour de force. We really appreciate it.
Thank you.